Take it away, Shrishti.
Thank you so much, Lawrence, for the wonderful introduction. And thank you, Sintika, for giving me the chance to talk about my work today. So it is titled Designer and Targeted Contrast Agent for Photoacoustic Imaging. And before we begin, I'm just going to talk briefly about the common imaging modalities that are currently present. So for clinical applications, there are a lot of imaging modalities currently present that have been translated and are in extensive use, and they range all the way from X-ray to MRI to PET and CT. And all of these imaging modalities have their certain advantages, and they bring some information onto the table for clinical studies. As imaging modalities are developing, there is another imaging modality that's emerging onto the market, which is photoacoustic imaging for both preclinical use and currently being translated for clinical uses through as there is one instrument currently available that uses the principles of photoacoustic imaging. And what photoacoustic imaging brings to the market is, let me just take my pointer, is something is just functional and molecular imaging. So as you can see in this example over here by Weber, you can see the extent of vascularization in these pictures or angiogenesis. And these are some of the capabilities that photoacoustic imaging currently offers. So it is a near imaging modality and is being used currently a lot and being developed in the preclinical settings. And this is the imaging that we'll be talking about today and a contrast agent development for PAI. So just to give a brief introduction to photoacoustic imaging, what it involves is there is a light, a pulse laser light that is that irradiates a tissue or a chromophore. This is an optical absorber. The optical absorber absorbs the incident photons, and then it expands, undergoes a thermoelastic expansion, as you can see over here. And that results in the generation of pressure waves, and that causes the generation of sound signals or ultrasound signals that are picked up by an ultrasound transducer. So that is the main principle of photoacoustic imaging, and the image is reconstructed, and you do get a lot of functional information about the organ or the tissue that you are trying to image. So moving on, photoacoustic imaging uses two different types of chromophores. The first one is endogenous chromophores, different parts of the body wherein melanin, hemoglobin, whether oxygenated or deoxygenated, collagen and lipids are great optical absorbers, and they can absorb the incident photon from the irradiated laser. And because of that, these particular components are used commonly as a chromophore in photoacoustic imaging. But they do have certain limitations. There's a limitations on the biological process that can be studied because these are endogenous chromophores that are available. So there might be sites wherein they are less in number and do not produce a good enough signal or a good enough image. And sometimes they can act as a strong background, which masks out the molecules of interest that you want to study, whether it is a targeted drug, etc. There are also exogenous chromophores in terms of different types of cyanin dyes, like indocyanine, which is already an FDA-approved dye, metallic nanoparticles, and there is a plethora of contrast agents now being developed, for photoacoustic imaging. The main advantage of exogenous chromophores is that they increase the sensitivity of imaging, giving you images with better contrast, and they can help sometimes study tumors, wherein you have wherein they are devoid of high contrast endogenous agents. So there are certain advantages of using exogenous chromophores based on your study, based on what you want to study. So, but as we talk about photoacoustic imaging, there are some benefits of having exogenous contrast agents, as I mentioned earlier. The limitations of most of the current contrast agents, again, none of them are clinically available except indocyanine green, is a few limitations. There's a limitation with respect to size, especially if you're dealing with the aggregates of cyanine dyes, since that is a process of self-assembly. There can be biocompatibility issues based on the material that is chosen to make the contrast agent. Sometimes, most of the times, you require a nanocarrier encapsulation for adding targeting moieties onto the contrast agent. And I will mention briefly why targeting is important. Some cyanine dyes have low photostability, that is, they are unable to convert a lot of the optical energy into an ultrasound energy, and some of them have low photothermal conversion. So in terms of the commonly available contrast, I already mentioned that indocyanine green, or ICG, is a clinically available contrast agent. It's already been FDA approved. So ICG has been the most commonly used contrast agent for photoacoustic imaging, and several formulations of ICG has been developed. As you can see over here, there are formulations when ICG is encapsulated within the liposomes. There are formulations wherein they are encapsulated with polymers or metallic nanoparticles, etc. The only issue is sometimes there's a low loading content for these formulations. So you have a low dose of ICG reaching the target site, which can limit your contrast that you want within the image. And this ICG also has a low photothermal stability. So that's why people have been trying to develop more formulations of a contrast agent. And one of the formulations that we work with extensively is J aggregates of ICG, or indocyanine green. In this ICG, the dye is incubated in different experimental conditions, especially at elevated temperatures, to result in a self-assembled structure which is held together by electrostatic interactions. It's end-to-end arrangement of these monomeric dye, and they cause stacking. And this stack-like structure is known as J aggregates. And one wonderful thing about after they aggregate is ICG has a very high optical absorption, just the monomeric dye, at 780 nanometer. But upon aggregation, it shifts in the near infrared range and has a very high optical absorption at 895 nanometer. But there are several potential issues with the current ICG-JA J aggregate formulations that are present. Most of them require a nanocarrier to add targeting moieties and for size control, because, as I said, this is a process of self-assembly. So you need to encapsulate it within the nanocarrier for size control. So there's no formulation out there that has intrinsic size control. Because of that, you require additional downstream processing, and that increases the complexity of the preparation methods. So this is where my work comes in, when we prepared in ICG-J aggregates, but we added another ingredient to it, which is azide modified ICG. And I will tell you the importance of the azide groups later. But basically, we took ICG, the dye, monomeric dye, along with ICG azide, and in the monomeric solution, we incubated these two dyes at different temperatures, salt concentrations, at varying times with various ratios of ICG azide to ICG. All of the details on this is given in the publication linked in the presentation. And after incubating them at elevated temperatures, which was 60 degrees Celsius, we obtained the same kind of J aggregate staggered structure that I mentioned in the previous slide, but in this one, there were azide groups incorporated into it. And we noticed that we call these structures JAS, and we noticed that JAS had the same optical absorption at 895 nanometer that is inherent of the J aggregates of ICG. And we confirmed the formation of these aggregates by seeing a color change that is pretty common when this dye ICG undergoes aggregation. So monomeric ICG is lighter green in color, and we saw a darker green color upon the formation of JAS particles. Followed by that, we characterized the presence of the azide group. We wanted to make sure that the azide group was incorporated within these aggregates that were formed. And for that, we performed FTIR. On the N3, or the azide group, has a very strong band that is shown in the FTIR in the 2100 centimeter wave number. And that is what we observed, as you can see over here, when compared to the control ICG-J aggregates. We also measured the zeta potential, or the surface charge of the particles. And we did see that JAS particles were slightly less negative than the ICG inherent J aggregates, which we believe could be since the azide group is slightly more positive than the indocyanine green dye. After that, we measured the average size of these particles, and they were in the micron meter range. As I said before, there's no intrinsic control since this is a process of self-assembly. And because most of the JAS particles that we obtained, their average size was in the 1.3 micron meter range, as confirmed by the SEM pictures over here, when you have a polydispersity and the average size remains in the 1.2 to 1.3 micrometer range. But after that, we wanted to achieve something known as, which was the intrinsic control of the size of these aggregates without encapsulating them within a nanocarrier. That was one of the main aims of this study. And so we started doing step-by-step optimization studies. Again, all of it is linked in the publication. And by following these optimization studies, we took several parameters into consideration, being the total concentration of dye that we worked with. We varied that along with the salt concentration. In our case, the salt we chose was potassium chloride. And salt was a cationic salt was chosen, since we do know that salt affects the kinetics of aggregation. And these are anionic dyes, or negatively charged dyes. So we ended up choosing KCl as our first salt for optimization. And we varied the salt concentration along with the total dye concentration, the time of incubation, and incubated all of these solutions at 60 degrees Celsius. And after that, we observed that varying the dye concentration at 14 hours, after 14 hours of incubation at 60 degrees Celsius, we saw the same sort of 895 nanometer peak, but the size was smaller. So effectively, if we lowered down the KCl concentration to 0.1 millimolar, we observed that the average size of the aggregates was lowered down to the 220 to 250 nanometer range. And when we increased the salt concentration, we observed there was a slight increase to 600 nanometer. And at the highest salt concentration, which was the formulation we used before, we were obtaining aggregates in the micron size range. And just to confirm whether we were truly seeing nano-sized aggregates, we did some SEM imaging, and we did observe that we obtained particles with an average size of 250 nanometer. So now we move on to an audience poll. Lawrence?
Apologies for that. Yes, thank you, Shrishti, for this great talk so far. We're just going to take a quick break here and ask that you in the audience tell us a little bit about the research that you're doing. We just have two quick poll questions for you. Just going to launch them right now. And so just take, if you wouldn't mind just taking a few minutes to feel these out. We're just going to ask first, what type of work are you currently focused on? And you can select multiple, whatever applies. So this can include contrast agent development, cancer, tissue engineering and regeneration, neuroimaging, developmental biology, therapeutic monitoring, molecular imaging generally, or other. And the second question is more along the lines of what you're using right now in your research, whether you're currently already using photoacoustic imaging. And your options are, yes, I use photoacoustic imaging through a custom-built system. I use a commercial system. No, I don't use it yet, but I would like to, or any other answer there. So yeah, if you don't mind just taking a few moments for this. I see we have quite a few answers already, so just give it another 20, 30 seconds, and then we're just going to move right on. Perfect, just another 10, 15 seconds. Thank you everyone for participating in this. It really is helpful for us to make sure we target the right webinars and we have content that is relevant to people who are interested. Perfect. And Shrishti, I'll hand it right back to you. Yeah, excited to hear what's next.
Okay. So I mentioned that we used ICG-JA, and this is where it's going to come in use, is why did we do that? It is to be able to build a platform on which we can add targeting molecules so that we can study a specific disease, a specific tumor, etc. So for the purposes of this study, we used a very common peptide known as RGD, which can target integrins that is present on most cells. And that was just to prove for the principle of proving this particular study. But there are different targeting molecules available. And the good thing about the contrast agent platform that we developed, as I will show you in the next slide, is that we are able to attach different targeting molecules onto the platform, making this truly a designer platform. So if you want to target something such as breast, we can attach HER2 molecules onto the platform and target a breast tumor, a HER2 positive breast tumor. So there are several targeting molecules to choose from, as you can see in this particular image over here. And that was our intention, to be able to create a platform that can, onto which we can attach any of these targeting molecules. So just to go back onto our JAS particles, I mentioned that they have these N3 or azide groups present. And these azide groups can be attached, can be functionalized with DPCO biotin groups. This is a part of click chemistry, copper-free click chemistry, and this is a very common chemistry technique used. And after the attachment of biotin onto a platform, we can attach streptavidin. This is a pretty common conjugation that's used by most researchers. And after that, onto the streptavidin platform, since it has 3 extra binding sites, the protein itself, you're able to attach any biotin-related molecule onto this platform, whether it's a biotin-related antibody, which can target a specific organ or tumor, whether it is a biotin-related aptamer or peptide. In our case, as I told you, we used a model peptide known as RGD. We are able to achieve this onto this JAS platform. And after, we wanted to make sure that even after several steps of functionalization, we do not lose any of the optical properties. And we did notice that the 895 nanometer peak is retained even after several steps of functionalization and filtration and purification of these particular particles. And after that, we measured the zeta potential or the surface charge of these particles, and noticed that on the attachment of streptavidin, since it is a 55 kilodalton protein, we observed a change in the zeta potential towards the positive end. It moved, so our particles moved from negative 57 millivolt down to 13 millivolt. And on the attachment of RGD, since it's such a small peptide, we obtained there was a non-statistical difference in the surface charge. We also characterize these particles just by using EDS. And after doing that, we were able to get a weight percent of the molecule of the elements present within the particles. So for the strep jas particles, when they're coated with the protein, you of course have a higher percentage of carbon, nitrogen, and oxygen when compared to jas particles, which have a lower percentage. So we were able to confirm the presence of streptavidin by using these techniques. Now, since the targeted platform has been made, we wanted to make sure that these particles are stable in vitro in different physiological media. And for that, we measured the change in their optical absorption at 895 nanometer while incubating them for an extended period of time in different media, which is media with serum, and 100% serum at 37 degrees Celsius. And for the jas particles, which are the micron-sized one, we did notice that there was no change in the optical absorption for up to 48 hours. For the nano-sized jas particles, which are named as NJAS in the nano-sized range, we did observe a little bit decrease in the optical absorption, maybe of 10% for 24 hours after incubating it in 100% serum. But overall, the particles remain stable in different physiological media. We also did an optical stability in whole blood just to make sure that they do not degrade down in whole blood, since this will eventually be a contrast agent that will be used for in vivo studies. And we observed that the jas particles, the micron-sized one, had about a 50% degradation after 8 hours in whole blood. The nano-sized one, the degradation was lower, which was around 20%. So the particles were 80% stable for up to 24 hours. The RGD jas particles, which is coated with streptavidin and the RGD targeting molecule, remain stable for up to 24 hours. So this coating of the protein on top of the jas particles really do give them a stability in whole blood also, which prevents their degradation. And we did expect this, and we were very happy with these results. We also wanted to make sure that these particles were something that, could be stored and resuspended for future use. So we did lyophilize the particles, and both jas and NJAS particles. And before drying and after drying, we did not observe a significant difference except for the nano-sized particles, which there was an 80 nanometer increase. But we are working on the stability right now, and we observed no difference in the zeta potential. Next, we performed in vitro photoacoustic studies by using a lab photoacoustic setup. Within the setup there, we used a 35 megahertz single element Olympus transducer, which is shown over here, immersed in deionized water. This is a well containing deionized water. And then within the well, there's a 96 well plate containing all of the samples embedded in agarose, along with we also use a Optotec wavelength tunable pulse laser, which is shown in this figure over here, which was pointed oblique onto the samples. And this was done to measure the photoacoustic properties of the samples. And we measured the photoacoustic signal that was generated by 3 different formulations: the micron-sized jas particles, the nano-sized NJAS particles, and the RGD attached particles. And we did these studies at 3 different concentrations, which were pretty low total dye concentrations. And as you can see over here, the RGD jas particles, we were thinking the coating with the protein might increase the photoacoustic signal. And we did observe that, especially at 25 micromolar of free dye. But overall, all 3 formulations gave us a pretty strong photoacoustic signal. And after that, we wanted to make sure that the optical absorption profile and the photoacoustic profile of the different formulations were similar. And we charted or did a wavelength scan or at every 5 nanometer for the photoacoustic profile. And we did observe there is a 5 nanometer difference, which I'm currently not sure why, but it was closely in agreement with the PA profile was closely in agreement with the absorbance profile. The next thing that we wanted to do was we wanted to see the PA signal when compared to whole blood, since this will be injected in vivo. And for that, we measured the normalized PA signal. The signal is normalized with respect to India Inc. of just monomeric ICG-JA. The two different formulations that I talked about, along with the targeted RGD formulation against blood. And as you can see, the targeted RGD-JAS formulation has a PA signal which is 2 times excess of that of blood. So we were pretty confident that this can be a contrast that can be injected and you will be able to see it against blood. And after that, we did some in vitro cell targeting studies. In this, we expected the RGD JAS particles to be attached onto the cells itself and give us a strong PA signal. So this is a 2D PA scan wherein the stage was moved and the cells are grown on a glass slide. And as you can see over here, the cells were incubated with the JAS particles, and there's an M region scratched onto the slide. And the slide was repeatedly, the JAS particles was repeatedly washed out after incubating, after the cells were incubated with it for a few hours. And the PA signal that is obtained from this particular slide was really, really low because these particles do not attach to the HeLa cells. Whereas in the case of RGT JAS particles, there is a region that was scraped out to act as a background or a control, but the rest of the regions containing the cells incubated with RGT JAS particles, and even after repeated washing, you were able to see a pretty strong signal. That's because, and we were able to confirm the presence of the RGD molecules, which targets the cell integrin. Moving on, after completing our in vitro studies, we moved on to our in vivo studies, and more details about the in vivo studies are definitely given in the publication link below. And the in vivo studies were done in collaboration with Photosound and used the TriTom imaging system. The in vivo studies were done on 2 new mice, and they were inserted into the sample chamber over here. There was a baseline PA scan obtained of the animal before the injection of ICG, which was used as a control, and RGD JAS was the particles used for study in this in vivo study. And before these 2 injections, there was a baseline scan obtained. After that, ICG was injected into 100 microliter of ICG was injected, followed by a 50 microliter saline flush. And these images of the trunk of the mouse was obtained. You have the coronal images and the axial images over here. And as you can see, over 90 minutes, we were able to have some enhanced visualization of the spleen and some part of the liver, but the intensity decreased after 90 minutes. But for the RGT JAS particles, as you can see, the moment it was injected at t is equal to 0 minute, you had an enhanced visualization of especially the liver, the spleen, and the superficial thoracic vessels. And this we could see these components for up to 90 minutes. And in the paper, as you can see, it was up to 120 minutes after injection. So there is a clear difference in the intensity of both the ICG, which is the current gold standard, and the RGD JAS particles. After that, we performed spectral unmixing. Over here, red indicates oxyhemoglobin, green is the blue is the oxyhemoglobin, and green is the RGD JAS particles. But the biodistribution of this contrast agent seems to be in the blood-rich organs. As you can see in this 3D video, the contrast agent accumulates within the liver and the spleen, which are blood-rich organs, but it can clearly be detected against oxy and deoxyhemoglobin. And the contrast agent stays in circulation for up to 90 minutes, as you'll be able to see in this video over here, and is able to give enhanced visualization of these blood-rich organs. And we were able to calculate the contrast to noise ratio of just our current gold standard ICG, which was 1.59, compared to our contrast agent RGD-JAS, which was 2.42. And also the circulation half-life was quite enhanced for these RGD-JAS particles. The details on these calculations are again given in the publication. So just to conclude, we were able to create a contrast agent that did not require nanocarrier encapsulation for its size tunability. We were able to tune the sizes from the micron size all the way up to the nano size range. This is again a reproducible and a very robust method of making the contrast agent. It also has clinical translation potential since it is composed of an FDA-approved dye, which is ICG, and has a strong optical absorption in the near-infrared range at 895 nanometer, and has multiplexing properties wherein you can attach different targeting moieties, making it a designer contrast agent. Of course, this work would not have been possible without the people behind it. I would really like to thank my advisor and my current PI, Remy Venziano, for all of his efforts behind this work, and along with Parag Chitnis, with whom we performed most of the in vitro photoacoustic studies, and he is the expert in photoacoustic over here, and his graduate student, Giovanni, who performed the in vitro studies. I would also like to give a special thanks to Dr. Dylan Lawrence, who performed the in vivo studies in conjunction with Photosound, and all of the wonderful people that I work with. And that would be the end of my presentation, and I will take all questions now.
Thank you, Shrishti. Thanks for the fantastic talk. And as mentioned, we're just going to jump right into the Q&A session. So please take a moment to type any questions you might have into the question and answer box at the bottom. And we have a few already, so we're just going to get started on those. And feel free to add more as we go along. So Shrishti, just to start off, obviously ICG is an FDA-approved agent, but how does adding azide and targeting molecules affect the potential toxicity of this agent? And for example, your studies show that this stays in the body for much longer than ICG. So how would you say that plays into toxicity, and do you foresee issues with FDA approval?
So we are till now we haven't we are performing the cytotoxicity studies. We've done in vitro toxicity studies in the lab, but the in vivo toxicity studies are still going on. I would potentially think it doesn't have a lot of effects on the body because it is composed of ICG in itself and its azide modified form. But the true histology studies of it is being done, and I would not be able to give you a correct answer until those studies are not performed.
Perfect. And the next question, there's actually two questions on this topic, we may have missed this, but does the protective coating also protect against photo bleaching, which is common with ICG? Or in other words, can you comment on the overall stability of the particle?
The overall stability of the particle, if irradiated with the laser directly, okay, without any collimate in between, is up to 2 minutes, and this is already given in the paper in the SI. But we do believe that coating it with the protein does decrease its the photo bleaching. Also, the aggregates themselves are inherently more stable optically and less susceptible to photo bleaching when compared to just ICG dye. So we have shown in a few of the results in the SI that yes, they do. There is less photo bleaching, and they do remain stable when compared to ICG. because of
Perfect.
the coding of the protein.
Excellent. The next question, you showed that there's a small shift in the spectra between the spectrometer readings and the photoacoustic
Yes,
scan, something along the lines around 5 to 10 nanometer difference.
fine.
And can you hypothesize on maybe why you think this must be the case? Could it be that the normal ICG shift that comes with concentrations is still playing a role somehow?
It can be the case. We did try to control the concentration as much as we could, but inherently it's very difficult to determine the concentration of the aggregates because we have to dissolve them in using a surfactant, then measure their concentration of free dye present, equivalent free dye. So the concentration can play a slight role, and that could be one of the reasons for the slight shift. But but I think that could be one of the reasons, but I wouldn't be able to comment more than that. So we are still working on these studies.
Perfect. And we have one more question about your in vivo study. If you don't mind going back to that slide,
Yes.
looking at the in vivo mouse images, one back from there, I think
Yes.
they're talking about these ones. Yeah, they're talking about the timelines from 0 to 90 minutes. And how are the images scaled to each other? Because like it looks like the RGD-Jazz images are brighter right from the get-go from 0 minutes, which understanding 0 minutes is after the injection, not the baseline. But is this like a factor of 2 times brightness compared to ICG, 3 times? Because just looking at it like here, it looks like they're all the same brightness across the RGDM and across ICG.
So do you mean the brightness when compared to ICG? That's like at least
Yeah, are they comparable or are these scaled differently? Or
yeah, no, they're comparable. They're scaled the same way. Okay. And yes, the moment after the injection, you do see the contrast flowing into the liver and the spleen and accumulating over there over time. And so they are scaled with respect to each other. And there, of course, there's an excessive 2 times higher contrast when it comes to RGD-JAX.
thank you. I actually think that's all the questions we have at the moment. So I guess we're going to wrap up and end the session today. As mentioned at the start, we will be answering or transcribing all of these questions that were answered on paper, and all of it will be available online along with the recording, as well as with
Lawrence,
the
there
slides
seems to
themselves.
be another question in the chat.
You're correct. Another one just popped up. Yeah, we can totally go answer that. Um, It's asking, how does the photoacoustic signal look across the 120-minute imaging time? So does it change across 120 minutes?
It does change across 120 minutes. It's lower than the 90-minute time point, definitely, because the contrast is being metabolized by the liver. That is the metabolic pathway of ICG. So we are hoping that the contrast follows the same metabolic pathway. So at 120 minutes, the signal is reduced, definitely. So it does look different. We did
Sounds
not
good.
image more than 120 minutes because we could not keep the mouse in under anesthesia for so long. So we do not know the exact time wherein the contrast leaves the bloodstream or is completely metabolized. But at 120 minutes, we do see a stark decrease.
Thank you. Well, if there's no further questions, I'd like to thank Shrishti for her presentation today, as well as take for time to address these questions. And I trust that we've been able to provide all of you with some relevant information to the studies you're conducting back at your own labs. If in the days and weeks to come, if you have any further questions about the work that was discussed today, or questions about any of the equipment, including the TriTom or photoacoustic setups, I encourage you to feel free to reach out to myself. I can also connect you with Shrishti. You can find me at lyip@syndica.com, or just on our website, you can find our contact information as well. And I'd love to welcome the opportunity to discuss your specific research goals or how any of our equipment might be able to help you enhance your research. And so thank you again for all of you for taking your time out of your day to attend the session. And thank you again for Shrishti for the great talk. And we look
Thank
forward
you.
to seeing you at a future Syndica event. Have a great
Yes,
day.
of course. Thank you.